Good day, everyone. We're the reporters of Group 10 from STEM 12 Algin, and today we will explore the interesting world of nucleic acids. But before we proceed, let us first identify our learning objectives for this lesson. After this discussion, the learners should be able to do the following. First is to understand the definition of nucleic acids. Second is to identify the two types of nucleic acids and their functions. Third is to describe the structure of nucleic acids. And lastly is to differentiate the two types of nucleic acids.
My name is Ron Andre Yape and I'm here to present what is nucleic acid. Nucleic acid is a naturally occurring chemical compound that is capable of being broken down to yield phosphoric acid, sugar, and a mixture of organic bases. So, nucleic acid are complex molecules found in living organisms, composed of nucleotides unit. These nucleotides consist of phosphate group, a sugar molecule, and an organic base. When nucleic acid break down, they yield phosphoric acids, sugar, and a mix of organic bases, including purines and pyrimidines. This breakdown is essential for various cellular processes and the transfer of genetic information. Next, nucleic acids are the main information-carrying molecules of the cell, and by directing the process of protein synthesis, they determine the inherited characteristics of living things. So nucleic acid, including DNA and RNA, are essential for carrying genetic information within cells. Through their structure, these molecules guide the process of protein synthesis. This means that the information encoded in nucleic acids dictates the sequence of amino acids in protein, which are crucial for the structure and function of cells. As proteins contribute the various aspects of an organism, traits and functions, nucleic acid play a fundamental role in determining the inherited characteristics of every living thing.
Nucleic acids are essential biomolecules that plays a crucial role in the storage and expression of genetic information of the living organisms. There are two types of nucleic acids, deoxyribonucleic acid and ribonucleic acids, which is DNA and RNA. And here are some functions of the nucleic acids. Number 1. Nucleic acids are responsible for the protein synthesis in our body. Number 2. RNA is a vital component of the protein synthesis. Number 3. Loss of DNA is linked to many diseases. Number 4. DNA is an essential component for the transferring of genes from parents to offspring. Number 5. All of the information of the cell is stored in the DNA. And number 6. DNA or DNA fingerprinting is a method used by forensic experts to determine paternity. It is also used for the identification of criminals. In summary, nucleic acids are fundamental to life as they carry and transmit genetic information and are involved in the synthesis of protein, which are essentials for the structure, function, and regulation of cells and organisms.
What is DNA? DNA is a group of molecules that is responsible for carrying and transmitting the hereditary materials or the genetic instructions from parents to offsprings. DNA is known as deoxyribonucleic acid. It is an organic compound that has a unique molecular structure. It is found in all prokaryotic cells and eukaryotic cells. DNA has a unique molecular structure because it carries genetic information in the form of genes and acts as the genetic material for a variety of creatures. Because of its double helix structure, it is less reactive. The shape in its environment has the least impact on it. DNA is also capable of self-replication and creating an identical duplicate. During cell division, the molecule can replicate itself because of its special shape. The DNA helix splits in half to become two single strands when a cell is ready to divide. Two new double-stranded DNA molecules, each of which is a copy of the original DNA molecule, are constructed using these single strands as templates. Deoxyribonucleic acid is a polymer composed of two polynucleotide chains that coil around each other to form a double helix. So when we say polynucleotide chain, it is a polymer that composed of several nucleotides linked together. It also means that they are connected together that form a double-stranded DNA. So the DNA is the polymer carries genetic instructions for the development, functioning, growth, and reproductions of all known organisms and many viruses. Who discovered DNA? Johannes Friedrich Nietzsche was a Swiss physician and biologist. He was the first scientist to isolate nucleic acid in 1869. He also identified protamine and made a number of other discoveries.
We have three types of DNA. So the first one is aDNA. It is a right-handed double helix similar to the bDNA form. All dehydrated DNA takes an A form that protects the DNA during extreme conditions such as desiccation. So protein binding also removes the solvent from DNA, and the DNA takes an A form. And for the second type, we have bDNA. So it is the most common DNA conformation and is a right-handed helix. The majority of DNA has a B-type conformation under normal physiological conditions. And for the last type of DNA, we have zDNA. And zDNA is a left-handed DNA where the double helix swings to the left in a zigzag pattern. It was discovered by Andres Wang and Alexander Rich. It is found ahead of the start site of a gene and hence is believed to play some role in gene regulation. So that is the three types of DNA. Again, we have aDNA, bDNA, and the last one is the zDNA.
Now let us proceed to the structure of DNA. Imagine a ladder that is twisted. That is what DNA looks like. Deoxyribonucleic acid, or DNA, is described as a double helix or double-stranded molecule. This is mainly because of the hydrogen bonds formed between purine and pyrimidine, which are the nitrogen bases that hold the DNA strands together. According to Watson and Crick, DNA consists of two helical strands twisted around each other in a double helix, where each strand contains a chain of repeating units called nucleotides. These are the basic building blocks of nucleic acids. Each nucleotide is composed of three different components, namely sugar groups, phosphate groups, and nitrogen bases. Let us define each of them. First is the sugar and the phosphate groups. They link the nucleotides together to form the structural framework or strand of DNA. This backbone is composed of alternating sugar and phosphate groups that define the directionality of the molecule. On the other hand, nitrogenous bases, or also called as nucleobases, are nitrogenous compounds that form an important part of nucleotides. This is an essential component of nucleotides as it is one of the key components in the building blocks of genetic information. There are four types of nitrogen bases in DNA: adenine, thymine, guanine, and cytosine. Remember these types of nitrogen bases: A, T, G, and C. Take note that among the three components of DNA structure, sugar is the one which forms the backbone of the DNA molecule. It is also called deoxyribose, hence the name deoxyribonucleic acid. DNA also plays an essential role in the process of cell division. The DNA coils up, forming chromosomes, and each chromosome has a single molecule of DNA. It also plays an essential role in the process of cell division. For you to have deeper understanding about the structure of DNA, we provided a sample experiment. The photo shown is the DNA extracted from a banana. Usually, we will think that in order to see DNA, we need to use a microscope. But with this experiment, you can clearly see the DNA strands of the banana with your naked eye. This experiment is done by mixing smashed bananas with dishwashing liquid, rubbing alcohol, and table salt. The stringy substance that you see is the DNA. It has been removed from the millions and millions of cells that make up the banana. The dishwashing soap helps to break down the cell's outer membrane, and the salt helps to separate the DNA from the other materials in the cell. And because the DNA doesn't dissolve in alcohol, this substance helps the DNA clump together in separate layers.
So good day everyone, I am Altenica Sarangay and I'm here to explain the function of DNA. So the first one we have gene therapy. So gene therapy involves altering the genes inside your body cells in an effort to treat or stop disease. So sa madaling salita, ito yung gumagamot at nagpapatigil ng mga disease na dumadaloy sa ating pangangatawan. And the second one, we have DNA fingerprinting. So DNA fingerprinting ay sobrang common na para sa atin because ito yung madalas na ginagamit upang maging ebidensya sa korte and to identify bodies, track down blood relatives, and to look for cures for disease. So, DNA fingerprinting is a chemical test that shows the genetic makeup of a person or other living things. So, the third one, we have cellular metabolism. Cellular metabolism naman involves complex sequences of controlled biochemical reactions, better known as metabolic pathways. So, itong proseso na ito ay nag-aalaw sa mga organisms na patuloy na mag-grow at mag-reproduce, and also to maintain their structures and respond to environmental changes. So, for the third function, we have transcription. So, the goal of a transcription is to make a RNA copy of a gene's DNA sequence. Well, the fifth one is mutation. A mutation is a change in the DNA sequence of an organism. So this process, or this function rather, can result from errors in DNA replication during cell division, and also exposure to mutagens or a viral infections. And for the last function, we have replication. So replication occurs in three major steps. So the first step is the opening of the double helix and separation of the DNA strands. The second step is the priming of the new template strand. And the last step is the assembly of the new DNA segment. So that is the sixth function of DNA.
And now let's discuss the RNA. RNA stands for ribonucleic acid, and it is a nucleic acid present in all living cells that has structural similarities to DNA. Unlike DNA, however, RNA is most of single-stranded because it is more flexible and can fold into a variety of complex shapes, allowing it to perform a wide range of functions within the cell. This single-stranded nature allows RNA to participate in processes such as protein synthesis, gene regulation, and enzyme activity. An RNA molecule has a backbone of alternating phosphate groups and the sugar ribose, rather than the deoxyribose found in DNA. And an RNA is information in the form of base sequence na nagtatranscribe into messenger RNA, transfer RNA, and ribosomal RNA. An RNA is synthesized from the DNA through a DNA transcription. RNA performs a myriad functions in the cell. And RNA and DNA works together to produce proteins. Proteins are used as enzymes that carry out cellular functions. RNA itself can also be used as enzyme that mediates cellular chemical reactions. And lastly, RNA is also a basic component in the building of organelles referred to as ribosomes. If we go back to our past lessons, ribosomes are the site of protein synthesis, and their integrity is important for efficient protein production.
James Franklin Conrad was a biochemist famous for his research on viruses. He discovered that the nucleic acid centers of every virus particle were carriers of genetic information that manage viral reproduction. So hence Conrad's significant contribution to the biochemistry involved the discovery that the nucleic acid centers, specifically the isolated ribonucleic acid, within every virus particles carry genetic information crucial for managing viral reproduction. This has helped advance our understanding of how viruses replicate and provided key insights into the role of nucleic acids in genetic processes within viral organisms.
Going to the next part, there are three types of RNA, which are messenger RNA, transfer RNA, and ribosomal RNA. Let's start with mRNA or messenger RNA. Messenger RNA acts as the messenger between DNA and protein production. And it is a molecule that contains the instructions or recipe that directs the cells to make protein using its natural machinery. Messenger RNA serves as genetic template of a protein since DNA can't leave the nucleus. On to the next is tRNA or transfer RNA. Transfer RNA serves as link between the messenger RNA molecule and the growing chain of amino acids that make up a protein. And it is responsible for bringing amino acid together during translation to form the growing peptide chain that will become protein. Last but not the least is rRNA or ribosomal RNA. Ribosomal RNA molecule in cells that form part of the protein synthesizing organelle known as ribosomes. This RNA is a main component of ribosomes again, and rRNA combines with special proteins to form ribosomes, which then read mRNA to form proteins. And essentially, ribosomes contain RNA and help build proteins.
RNA ribonucleic acid is a molecule that plays a crucial role in the various biological processes, including protein synthesis, gene regulation, and the transmission of genetic information. The structure of RNA is similar to the DNA with a few key differences. The basic structure of RNA can be described as follows. Number 1. Like DNA, RNA is composed of monomers called nucleotides, and each RNA nucleotide has three components: adenine, uracil, guanine, and cytosine. Uracil replaces the thymine in the DNA. The sugar in RNA is ribose, which is an additional hydroxyl group compared to the deoxyribose sugar found in the DNA. A phosphate group is also part of the RNA molecular structure. While DNA typically forms a double helix, RNA is usually a single-stranded. However, in some cases, RNA molecules can fold back on themselves to form complex 3D structures due to intramolecular pairing. In summary, RNA is a versatile molecule with a single-stranded structure formed by nucleotides containing ribose sugar and different nitrogenous bases. Compared to DNA, its structure allows for a wide range of functions, including information transfer, protein synthesis, and gene regulation. And that is all for the structure. Next slide is the RNA isolation experiment. This photo shows the RNA isolated from yeast. It is an experiment that can help us gain better understanding of the structure of the RNA without needing a microscope.
Hi, I am Janelle Alvillariel, and for today I will be discussing the functions of RNA. So, for the first functions of RNA is that RNA create a protein. It is the primary role of RNA. Number two, functions as an adapter molecule in protein synthesis, which it serves as a link between mRNA molecule to the growing chain of amino acids to make a protein. Number three, it facilitates the translation of DNA into proteins, wherein mRNA carries the message out to the nucleus to the ribosomes that is located in cytoplasm, wherein the transfer RNA helps to translate the message to make a protein. Number four, it serves as a messenger between the DNA and the ribosomes because RNA converts the information that is stored in DNA. And lastly, RNA promotes the ribosomes to choose the right amino acids, which is required in building up a new proteins in our body. Now let's proceed to the differences and similarities of the DNA and the RNA. Let's begin to the differences of the two. The DNA is double-stranded, while the RNA is single-stranded. The DNA nucleobase is thymine, while the RNA is uracil. The DNA is found in the nucleus with a small amount of the DNA also present in mitochondria. In the cells without nucleus, DNA is found in nucleoid, while in the RNA forms in nucleolus and then moves to the specialized regions in the cytoplasm depending on the type of RNAs it formed. The DNA is made up of deoxyribose, while in the RNA it is made up of ribose. The DNA is also stores coded information while the RNA forms proteins and transfers them. In the DNA, it also can self-replicate while the RNA can have a multiple structure. In the DNA, it's read by polymers while the RNA is read by ribosomes. And lastly, DNA can last for a long time, but in RNA, it can't last for a long time. So now let's move to the similarities of the DNA and the RNA. DNA and RNA have a nucleotide building blocks. They also contain adenine, guanine, and cytosine. They both have a large biological polymers, and also the DNA and the RNA consist of sugar, nitrogenous bases, and a phosphate backbone. And that's all for our report. Thank you for listening and have a good day.